Hello everyone, welcome back to RPIIT Academics. I am Avika, working as an assistant professor in RPIIT Technical and Medical Campus, Bastada, Karnal. Today we are going to discuss about the peripheral blood smear or preparation and staining of peripheral blood smear. So for that we have to know about the blood film. So what is blood film? It is a thin layer of blood which is smeared or which is mounted on a microscope slide and then stained in such a way to allow the various blood cells to be examined microscopically. Basically, we examine the blood cells through microscope by preparing a blood film. It can be of two types. First is thin film and thick film, as you can see in this slide. Now, what is the aim of preparing the blood smear or peripheral blood smear or blood film? These are usually examined to investigate hematological problems. What type of hematological problems? Like disorders of the blood, and to look for the parasites within the blood such as malaria and filaria. So for that we have to prepare the blood films and then we investigate or we examine them. After that, examination of thin blood films is important in the investigation and management of anemia, infections and other conditions which produce changes in the appearance of blood cells and differential white cell count. A blood film report can provide rapidly and at low cost. What does it provide? It provides the useful information for the patient's condition or about the patient's condition. Now, if we talk about the types, it is of PBF. It is of two types. First one is the thin and the second one is thick blood film. So, main basic steps to prepare the film are preparation of blood smear and then fixation of blood smear, after that staining of blood smear. First of all, we do prepare the blood smears, dry them and then fix them. After then we stain them for the different types of cells to be recognized. Here you can see, first of all we will place a small drop of the whole blood on a clean slide and we will take the spreader, means second slide, over it we will take it in between 30 to 45 degree angle. We have to take an angle of 30 to 45 degree. While maintaining contact with the bottom side, we will pull the slide back to contact the drop, which will spread by capillary action. We will pull this towards the back of the drop. Now we will maintain the contact with the bottom slide and push the top slide in one motion to produce the thin layer or to produce the smear. Now, first of all, if we talk about the thin film or thin blood film, it can be prepared from the anticoagulated blood which is obtained from the puncture of vein or from the finger. If we prick our finger, then free flowing blood is taken, or we can take the blood, anticoagulated blood, which is obtained from the vein puncture. So basically three methods or three techniques are here. First is slide method, second one is cover glass method, and the third one is spin method. So, what is slide method? First of all, on a slide, we will place a drop of blood in the center of clean glass slide on one side, that is 1 to 2 centimeter from the one end. Now we will place another slide, that is called spreader, with smooth edge at an angle of 30 to 45. It should not be less or more than that, otherwise our film will not be proper. Now we will move the spreader backward while contacting with the blood drop. Then we will move the spreader forward rapidly over the slide. A thin peripheral blood film is thus prepared. Now we will dry it and then stain. Next is cover glass method. We will take a cover glass slip and we will put one drop of blood over the cover glass slip. Now we will place another similar cover glass slip over it in the crosswise direction with one side containing drop of blood facing down. So now we will pull the cover glass quickly from it and now dry it and stain it. After that we will mount it with a mounted film slide down on a clean glass slide. After that, third method is spin method. It is automated method. We will place a drop of blood in the center of glass slide. After that, we will spin this glass slide in speed centrifuge. Now, blood will spread uniformly in the centrifuge. After that, we will dry it and stain it. Secondly, thick blood film is prepared by detecting blood parasites. This is prepared for detecting the blood parasites such as malaria and microfilaria. For this, we will place a large drop of blood in the center of clean glass slide and now we will spread this in a circular area of 1.5 centimeter with the help of a stick or end of another glass slide. Now we will dry it and then stain it. You can see that we are putting here a blood drop. After that we are taking another spreader or stick to prepare the film. You can see the bad and good film which are preparing. You can see uneven or bad film and a good one. Now, if we talk about the qualities, what are the qualities of a good blood film? It should not cover the entire surface of the slide. The whole surface must not be covered. It should have smooth and even appearance. Its appearance must be smooth and fine. It should be free from waves and holes. There should not be any holes or air bubbles. It should not have irregular tail. Its tail must be regular. It should not be irregular. Thank you and have a nice day. If you like the video, please share and subscribe our channel. In the next video, we will talk about the fixation and staining of the glass slide or staining of the blood smear. Thank you.